Thank you for coming. We are going to talk about immune repertoire profiling and phenotyping of T and B cells. And probably I don't need to give introduction in this audience. You know more about adaptive immunity than me. And I am more technology guy, so that's why I'm going mainly to talk about technological aspects, how to do this type of assays and how to analyze this data. But in general, a list of the all typical application of adaptive immunity for organ transplantation, cancer, for autoimmune disease, infection disease is right now constantly growing in the wake of immunotherapy. Right now, immunologists start to be very critical part even for completely known area of application of human disease like cardiovascular disease or neurological disorders, which is completely out of the range 20 years ago. And I would like to remind you how these TCR and BCR genes are developed in the course of recombination, VDG recombination, what you study in the college. And it's really what is interesting in this system, all T cell and B cell receptors and antibodies organized very similar way, where there is region shown here in on the top of these structures. It's CDR3 region, recognize epitopes presented by major histocompatibility complex or directly in the case of B cell receptors or antibodies. From genetic point of view, what is the most amazing here is the most complex genetic system created by nature. Complexity of T cells and B cells, which present constantly in our circulation, it's at least several 100 million of different T and B cells with completely different T cell and B cell receptor sequence. This complex genetic system is designed by nature not to recognize any foreign antigen or pathological condition and eliminate this type of the cells. So it's universal diagnostic and therapeutic system. The question is, we don't know really well how to use it for diagnostic until now and for therapeutic application. Definitely, all these new therapeutics, for example, adoptive cell therapy or CAR-T therapy, it's all of them derived from this model which created by nature. From technological side, the most important is really how we're going to analyze this complex population of the TCR immune receptors and try to characterize cells which express these T cell receptors. And I'm going to talk about technology right now. In order to facilitate these studies, we developed several assays, like Paul already mentioned. First of them, it's adaptive immune receptor or AIR assay, which allow you to profile all 7 chains of T cell or B cell receptors in single multiplex reaction. It's very simple assay, which allow you to generate very quantitative data in very short period of time and cost-effective fashion. We also developed, in the middle you see immune marker or genome-wide driver map expression assay, where you can profile 19,000 of protein coding genes or selected set of 500 genes which combine only highly validated T and B cell markers. And single cell assay, definitely here it's like dominating technology right now. It's 10x technology for T and B cell receptor profiling and expression profiling. We also have alternative technology, more cost-effective, where you can sort cells in 96-well plate and generate very similar set of the data using a user-friendly format without any instrumentation, and it's using technology which you commonly used in the lab. If you are talking about interplay between this technology, you need to understand that probably bulk profiling using AIR assay, like shown here on the left side, it's probably easier entry point in order just to generate quantitative data in a cost-effective way. For example, I routinely run four plates, 96-well plates in one day. So it's 400 samples. And it's really, you can generate really reliable data which you can trust in order to identify activated clonotypes. On the other spectrum, it's single-cell analysis. Single-cell analysis, if you try to use 10x technology, it's rather expensive, but it generates very extensive, rich information. You can see if you go to any presentation at this conference, you can see a lot of data from 10x genomics, and this information is very important. Cost-effective strategy to run the same assay in multiple plates, and profiling in immune cell fraction is somewhere in the middle. It's combined benefits of single cell technology with the bulk profiling. Paul already gave introduction to this technology. This technology is allowed to, ERA technology allowed to profile all 7 chains in one reaction. Primers designed only to amplify functional isoforms. We incorporate universal molecular indexes, which are very critical for analysis of this complex population, because you label each molecule in the population with unique barcode. And after the next generation data, you can exactly know how many molecules, starting copies of each molecule, are present in your biological sample, which is very cool, and it's integrated with MIXCR data analysis package, which is probably right now the best package for immune repertoire profiling. If you try to use this technology, and here's example, if you take whole blood or PBMC and try to run this assay using, for example, 50 nanogram of total RNA, you generate data like shown here on this slide. You can see here all 7 chains, having light chains for alpha, beta, gamma, delta for T cell receptor, and IgH, IgK, and IgL for B cell receptor. And you identify approximately 100,000 clonotypes from 50 nanogram of the total RNA. The problem is when you start to analyze this data. And this one is cool, because when we start to analyze the data, you try to repeat this experiment 3 times in replicates, technical replicates, and you will see practically very low reproducibility in triplicates. What is the problem? How to generate any quantitative data from these studies? And here's the explanation what's happened. For example, he showed results of 200,000 clonotypes for heavy chain of T cell receptor. And you see from 200,000 identified clonotypes here, you see practically all clonotypes are present. You see here it's on the y-axis, it's numbers. This one's number of molecules what present in 50 nanogram of total RNA. And you see majority of 999,000 presented single copy of mRNA molecule in the sample. So it's rather obvious for everybody from single copy of molecules you cannot generate any reproducible data. It's present or missing if you do in triplicates this experiment. Only 500 genes, clonotypes, present at more than 10 copies of mRNA in this sample. So it shows the problem with the working with the extremely complex population of RNA molecule which correspond T cell and B cell receptors. So if you have more than 10 molecules in this data, in this case, data reproducible. You show here differences between in triplicates. And it's illustrated if you take top clonotypes, few hundred, data very reproducible in wide range of RNA concentration between 25 to 100 nanogram, or even for microsamples. And here's the result for 30 microliters of the blood dried in the neoterics tips. For DNA, you also can generate reproducible data, but here's the limitation that you need to start from really big amount of genomic DNA, because genomic DNA, I would like to remind, present a single copy in every cell. RNA present at a couple hundred copies, so it's 100-fold at least more sensitive than DNA-based assay. So that's why it's genomic DNA profiling if immune receptor require more DNA than definitely RNA. How to identify activated clonotypes? This one's because, okay, you identify this 100,000 of the clonotypes. What to do with these clonotypes? Probably you're interested in clonotypes which are interact with or activated by specific epitopes. Or, for example, neoantigens, for example, in case of the cancer. So in order to illustrate how it can be used, here's some sort of longitudinal studies of vaccination. And here's, you can see on the top, it's 2 types of vaccines. It's triple vaccine, tetanus, diphtheria, and pneumonia. And you can see, and another one, shingles. And we collect multiple data points, purify RNA and DNA from these samples, and perform genome-wide expression profiling, single-cell analysis, and AIR assay. And here's like, it's in classical immunology book. Here's what's happened if you try to do expression profiling in these samples. And you can see in the 2 days after vaccination, you can have activation of group of the genes, which are all of them majority of them correspond innate immunity. It's very fast activation of innate immunity genes. In 1 week, you have activation of adaptive immunity, shown pathways shown here on the right side in the bottom. If you try to do RSA using DNA and RNA, and here's the result, it's rather complex image, but if you compare, for example, here it's heavy chain of immunoglobulins, you can see here on the DNA, you can see very clear activation at about 1 week after immunization. RNA, first notice RNA, it's rather obvious immediately from this image, it's at least 100-fold more sensitive than DNA, and which is rather obvious because plasma cells should generate a lot of mRNA and antibodies in order for this type of the immunization efforts. We can see also rather clear activation for T cell receptors. We don't see at the DNA level, so it's probably indicate not dramatic proliferation of the T cells in immunization, but on the RNA level, you can see activation of some specific T cell receptors. Okay, this type of the, if you try to, after that, just to convert these sequences of T cell receptors and predicted neoantigens, or in this case, it's vaccine antigens, we have nice collaboration with the company ImmuneWatch, and this company is probably the best software tool in order to detect epitopes, predict epitopes which maybe interact with specific T cell receptor. So we take this group of activated clonotypes, sequences of CDR3 region, and apply this program, which is machine learning algorithm. It's rather complex, but it's allowed to predict and demonstrate that practically all clonotypes we detect correspond only one protein, glycoprotein E, for zoster vaccine virus, which is show power of this strategy. Definitely it doesn't work for all T cell receptors because its current stage of the technology development prediction epitopes for specific T cell receptor sequences is rather in beginning, because we have right now only between 2,000 to 3,000 of validated interactions between specific peptide and specific T cell receptors. So this amount of information is not enough in order to build really very comprehensive, reliable model for prediction of the epitopes, but it's work in many cases. So in the case of vaccination, it's rather easy. You have multiple samples and you easily identify activated clonotypes based on the time course of activation. But what to do in some difficult cases? And here's the result of our collaboration studies with Professor Wilson Hudson from Emory University. And these scientists try to identify activated clonotypes in metastatic brain samples. So what they done, they purify T and B cells from metastatic brain samples and perform DNA analysis and RNA analysis using our technology. So you can see here overlapping clonotypes exist between both RNA and DNA. RNA is more sensitive, but the problem how to identify clonotypes if you have only one data point for each person. So we speculate that if cells, T cells or B cells are activated in in tumor environment, in many cases it can stimulate expression of these genes. So normalization of the RNA data to DNA data, actually to copy number of the cells, allow probably you identify, not in all cases, but in some cases, activated clonotypes which are stimulated by neoantigens and not correspond bystander T cells or B cells which are present in a specific tumor sample. So in this one, it's comparison between DNA and RNA data, and you see on the right side, you can see here it's IgH, heavy chain. You can see here a very clear picture. On the DNA level, we practically don't have any, you can see here it's like practically no any red color. So it means it's no dramatically significant abundant clonotypes if you just look at the DNA level. But approximately 10% of the clones, for example, look here, has very high level expression of the RNA. So if we have approximately 1,000-fold activation of the transcription in comparison with the copy number of genomic DNA for these overlapping clones, which allow you to identify, narrow down this list of few thousand clonotypes to something manageable number of B-cell receptor sequences for follow-up functional studies. The same is true for T-cell receptor, but here fold activation is up to 50-fold. Next step in any journey when you start to do all this type immune profiling, it's really to characterize phenotype of the cells. As Paul mentioned, we developed some assays. This one is focused assay for 500 of T and B cell markers, which are very heavily characterized in the panel of 40 immune cell fractions, and allow to differentiate between different type of naive memory effector cells for CD4, CD8, NK, Treg. And this type of assay can be used for immune phenotyping in the sorted cells or cell fractions. When we are talking about immune phenotyping, definitely dominating technology right now is 10x genomics, which allow to encapsulate cells in droplets after generate expression profile from the each cell and cluster this profile based on the similarity in expression profile and identify each cell type using some specific cell type specific markers, for example, markers specific for naive cells or memory cells. Actually, you can generate very similar data if you just sort the cells using the same markers specific for naive and memory cells. And after that, you can do expression profiling using this marker panel, what I discussed, or air profiling assay in order to identify all clone types which present in each cell fractions. In the middle, it's analysis using, you can sort the cells in 96-well format and generate very similar data, but for smaller number of the cells. And here's an illustration. We sort the cells in several fractions of naive memory cells and run assay directly from the cell without purification of the RNA or DNA, and both for air assay and for 5-marker TNB panel assay. And you can see on the left side, you can see expression of these 500 markers, which clearly differentiate different cell subtypes. And here's the set of immune receptor CDR3 sequences, which are specifically associated with each specific phenotype. Last probably slide would like to show you, it's like application of sorting of the cells in 96-well format. And what we believe in many cases, when you try to study, for example, activated cells, or for example, cells are activated based, these cells can be sorted, for example, using cell activation marker, for example, CD137, or try to study some sort of the exhausted cells. Cell number is really very low. It's very often it's only a few hundred cells. And in this case, it doesn't make sense to go to expensive platform which allow to analyze 10,000, 1 million cells. You need just to sort the cells in 96-well plate and generate all data like shown here, combination of full-length information for all heavy and light chain, expression of 30 markers, which and we provide this type of technology which allow to do it really in 96-well format. And I would like to summarize right now my presentation. We discussed development of air assay, which is profiling, comprehensive profiling in quantitative fashion of all 7 chains of immune receptors. We discussed detection of activated clone types based on the normalization of air RNA against air DNA profiling data. We also discussed even possibility to predict epitopes using immune watch detect algorithm, and some strategies for immune phenotyping of the cell using high-throughput 10x genomics technology, sorting cells in the cell fractions or in 96-well plate. And all these technologies, what we discussed today, available as the kits or custom services. And if you're interested to use this technology, please contact us. It will be our pleasure to help you in your research. Thank you.